So metabolic pathways of erythrocytes normally includes this Embden-Mialov pathway, which is also called anaerobic glycolysis. So this is for production of energy in form of ATP, and this actually there's just very low energy production because tricyclic acid cycle or Krebs cycle is better for energy production, but in this case glycolysis only can generate 2 ATP, which is very few, but of course by losing also 2 ATP, total 4 but net gain of 2. So glycolysis in fact for other purposes like use of sugar and so on, it is okay, but for us we are not going to talk about energy production because that one is a chemistry part, but we are going to discuss about glycolysis diversions pathways. Diversion pathways are very useful for RBC metabolism and RBC survivors or red blood cell survival, because RBC need diversion pathways for them to survive. So we are going to look at those diversion pathways and how important they are to the RBCs. In fact, we have 3 main diversion pathways. So you can see what diversion is like: diverting from original pathway. They're like switching to another new pathway from the main or the mother pathway. So that is diversion in this case. Now, those diversions include hexose monophosphate pathways. Probably you find it. You will actually sometimes find it with other names, but it is called hexose monophosphate pathway or HMP. There's also another pathway which is called methemoglobin reductase pathway of RBCs, and even the last one is called the Rapoport-Luebering pathway. So these are the 3 diversions from the main anaerobic glycolysis. So now you know how it works. Glycolysis, how it works. It starts with the glucose, which is of course acted upon by hexokinase enzyme by addition of ATP and generation of glucose 6-phosphate. Very simple technique, very simple pathway. You remember the whole pathway until you get pyruvic acid. So now let's not just concentrate on this long pathway, but let's look at these diversions from the main pathway. The first pathway is called hexose monophosphate shunt or hexose monophosphate pathway. It is diverted from glucose 6-phosphate. As I have just said it, glucose 6-phosphate is the second molecule in glycolysis, but from that molecule there is diversion. There is like switching to another pathway. So the pathway is called hexose monophosphate. So this glucose 6-phosphate, the action of glucose 6-phosphate dehydrogenase, it is oxidized to 6-phosphogluconates. So this second molecule of the original glycolysis is oxidized to 6-phosphogluconates by this diversion, by the catalysis of the glucose 6-phosphate dehydrogenase enzyme. So in this process, NADP is reduced to NADPH. NADPH is produced from reduction of NADP during this process of oxidation of glucose 6-phosphate. So you remember what you call redox reaction. That's where you find that one molecule is reduced along with reduction with the oxidation of another molecule, and then that is what happened. And of course you can see now, because there is reduction of NADP and production of NADPH, there will be also a chance to reduce glutathione. So glutathione is reduced by NADPH. Without NADPH, then glutathione cannot be reduced because it has been oxidized during some processes. Because you know that glutathione is antioxidant, it it normally it reduces oxidant in our body, and when glutathione is reducing oxidant, is also oxidized. So when it is oxidized, it becomes useless. So it will require NADPH in order for it to be reduced again. So this NADPH is very important, and it is generated from hexose monophosphate shunt. So glutathione get oxidized, of course, when it is reducing peroxide. Peroxide is an oxidizing agent, is oxidant. It can destroy cells because it can oxidize components of the cells, including proteins, lipids, and so on. So in this case now, peroxide, which is in the body, will oxidize glutathione when glutathione is trying to reduce it to water and oxygen. So now, therefore, hexose monophosphate pathway can detoxify peroxide, which oxidizes ferrous iron into non-functional ferric states, and peroxide can also oxidize and destroy proteins and lipids. So you can see now, by trying to remove peroxide, glutathione is oxidized, and it requires NADPH in order for it to be reduced again and continue its works. But of course, because of hexose monophosphate pathway, there will be continuous generation of NADPH ready to reduce the oxidized glutathione. So that's the function of hexose monophosphate pathway. So the second pathway, which is also a diversion from the main anaerobic glucolysis, is methemoglobin reductase. Methemoglobin reductase pathway, it is another type of pathway. So, but it works once ferrous iron is oxidized already, has been oxidized already, then hemoglobin has already changed to methemoglobin. So methemoglobin is useless hemoglobin, and because this action cannot be reversed by hexose monophosphate pathway, hexose can prevent it, but it can't revert it when it has already happened. So the requirement is the next pathway, which is methemoglobin pathway, so which is also involve NADPH produced from glucolysis when glucosidehydrogen 3-phosphate is converted into 1,3-biphosphoglycerate, reduces methemoglobin in the presence of methemoglobin reductase, right? Very, very crucial pathway. So this happened when glycerides, this called glycerides 3-phosphate, is being converted into 1,3-biphosphoglycerate. There will be also further reduction of NADPH, and then NADPH can also be able to reduce methemoglobin back to hemoglobin. Of course, NADPH will work as a coenzyme with methemoglobin reductase enzyme that is going to reduce methemoglobin. That is also another importance of some of the diversions pathway. So the last one is called Rapoport-Luebering pathway. Rapoport-Luebering pathway is another diversion pathway where a 1,3-biphosphoglycerate generated from this step, from the previous step of methemoglobin reductase pathway, this 1,3-biphosphoglycerate is diverted into another molecule. This is like isomerism, it's like diversion, but there's no change of any atom on it, but it's only like switching the position of phosphate. So this 1,3-biphosphoglycerate, it is still the same as 2,3-biphosphoglycerate. They're the same, only the difference is position of phosphate on the molecule. Here it is on position 2 and 3, and previously it was on position number 1 and number 3. So here now, because of this diversion by phosphoglycerate mutase, 2,3-biphosphogyrecerate is a very important molecule. So without 2,3-biphosphogyrecerate, there will be no oxygen delivery in tissues. So 2,3-biphosphogyrecerate assist in oxygen delivery in tissues and stabilize the oxygenated hemoglobin. So binding of this molecule 2,3-biphosphogyrecerate will switch or will shift hemoglobin dissociation curve or hemoglobin oxygen dissociation curve to the right. That is what we call right shift. Right shift is going to increase oxygen delivery in tissues. So that's all about the three EMP, embryonic pathway diversions that are very crucial for RBC metabolism. So the last thing that we're going to see just briefly, like in 2 minutes, is about heme biosynthesis. Also, it is very important, so I can't skip it. And then after we finish with it, we'll have done with everything we are supposed to know on hemoglobin metabolism. Other things will come later in other course and other lessons. So now, hemoglobin metabolism, of course, it's about heme metabolism, because I've seen hemoglobin and we'll still see it later in other courses. Heme metabolism is very important, or heme biosynthesis. Heme biosynthesis begins in mitochondria with condensation of glycine and succinate coenzyme A. So there will be in mitochondria, that's where it starts. So heme biosynthesis, it starts with condensation, that is of course combining two things by the loss of water molecule of glycine amino acid plus succinyl coenzyme A by the enzyme aminolevulinic acid synthase, and this condensation is going to produce aminolevulinic acid. Aminolevulinic acid, then it will migrate from mitochondria into the cytoplasm or cytosol, and in the cytosol, aminolevulinic acid will be converted immediately by porphyrinogen synthase, and it will become porphyrinogen. So porphyrinogen will be the second molecule from aminolevulinic acid in the cytoplasm. So then porphyrinogen will also go further deamination in the cytoplasm and is then converted, of course, by porphyrinogen deaminase into hydroxymethyl So hydroxymethyl is also found in the cytosol, but after undergoing deamination of porphyrinogen. So then hydroxymethyl by the action of uroporphyrinogen synthase, it will be converted into another molecule, which is uroporphyrinogen 3. Uroporphyrinogen 3 will be a molecule after hydroxymethyl. So it will be the fourth molecule in heme synthesis. So the fifth molecule will be a conversion or decarboxylation of uroporphyrinogen 3 and becoming coproporphyrinogen. Coproporphyrinogen 3 is the molecule which will be produced after decarboxylation of uroporphyrinogen 3. Then coproporphyrinogen 3 will be the fifth molecule in heme biosynthesis. So this coproporphyrinogen 3 will in fact migrate back into the cytoplasm, and it will be oxidized into protoporphyrinogen 9. So protoporphyrinogen 9 will be another molecule back into the mitochondria after oxidation of coproporphyrinogen 3. So then this protoporphyrinogen 3 molecule will also undergo further oxidation and become protoporphyrin 9 in mitochondria. So protoporphyrin 9 is the last molecule in heme biosynthesis, which will be, of course, also worked upon by pheroteratase enzyme. So pheroteratase enzyme is now able to bring ferrous ion or ferrous atom, that is iron 2, and it incorporates it into protoporphyrin 9. Then it forms heme. So heme is completed in the cytosol and is released into the cytoplasm, and then it combines with the hemoglobin in the cytoplasm, with the globin chains to form hemoglobin. So which means that heme is produced in various steps from cytosol, cytoplasm, from cytosol to mitochondria, back and forth, and then finally assembly of hemoglobin will happen in the cytoplasm. So then, very important, there's one point I want to point out, I want to mention. Some questions, sometimes they used to bring questions where they ask you, what is a good sequence of these types of molecules? Maybe they will just mix up everything and they will ask you to show a correct sequence of this molecule. So then I advise you, let me show you how I memorize them at first. First of all, there's one molecule called aminolevulinic acid. This is the first one. You should always know that the first molecule is aminolevulinic acid, which followed by PBG, porphobilinogen, where you find a B. B is found once here. Porphobilinogen is the second molecule, and then hydroxy is also a unique molecule. Hydroxymethylabilane is the third molecule, following porphobilinogen. So B should become 2, porphobilinogen, and also the fourth and fifth, they are almost the same. So you know what you call urine molecule. Urine specimen. For us, it is just a specimen, right? So urine is called uro. Stool is called copro, right? We have stool and urine. They're two different things. Stool is called copro and urine is called uro. So then it means that hydroxy is followed by uro, and then uro is followed by copro, which is the fifth molecule. And of course, after copro, there will be proto, and there are only two. There's one proto which is ended by gene, and gene means production, right? Or giving birth to something, production. And then gene, there's another proto, which is the last proto, which is ended by 9. It is just protoporphyrin without a gene. So that one is the last one. And finally, after that, there will be heme, right? So then it becomes very easy. So to memorize those 7 molecules, right? So you could say that there is aminolevulinic acid, porphobilinogen, hydroxy, the third one, then uro, it stands for urine, copro stands for stool, that is the fifth one. Then we are now at proto. So proto means proto, gene, then proto, the last one without a gene, and then heme. That's how you can memorize them. It's very simple, isn't it? Very simple. I think you should clap for me, because I've now shown you how you can memorize those things very easily. Thank you so much. And that's how I memorize them, right? Very simple. And I think I saw one question, and not even one. They can be different, but it is very important even to know. And if possible, try also to understand enzymes that are involved, even though we are going to see them in the lead poisoning, because lead poisoning we are going to come back to heme synthesis, because you know that lead poisoning can also interfere with heme synthesis. So maybe this time don't memorize these enzymes, but you are going to see them again in lead poisoning and in general megaloblastic anemia. Thank you so much for joining the session. We'll keep in touch. So tomorrow is the next one, of course. It will be about leukocytes until we finish this hematopoiesis and physiology of blood, and then we continue with other key points in hematology. God bless you, and we keep in touch.